Hello, and welcome to our project. We're going to be looking at how environmental factors impact pollen viability on a native plant in Northern California. My name is Erin Hall.
And I'm Jeanette, Moritz.
Alright, so as we know, climate change is impacting everyone globally, but specifically plants as well. And this is harmful in the fact that they make up 80% of the food we eat, as well as 98% of the oxygen we breathe, so they're very necessary. So the focus of my thesis, which this project is helping with, is how plants are able to cope with climate change, and they can do so in 3 different ways. They're able to migrate, they can exhibit adaptive plasticity, or they can adapt to their novel conditions they're presented with. So my thesis focuses on phenotypic plasticity and how it's presented in serpentine soil. So serpentine soil represents some of the harshest growing conditions for plants in the natural world. It's super porous, it has low nutrients, and has elevated toxic metals, so we're not entirely sure how plants are able to grow in such conditions. So the main research question is, do varying environmental factors have an impact on the amount of viable pollen produced in plants? So our study sites, we can see on this map here, the orange layer is the serpentine soil, and then in the north is our Marin site, and the south is Santa Clara. So the northern site, Marin, is known to be wetter, while Santa Clara is known to be hotter and drier. And we chose these sites as they house both serpentine soil, but also my study species, which is Antirrhinum vexycilocultum. It's a wiry snapdragon, and it's native to Northern California, and it's serpentine tolerant, so it can grow on and off serpentine soil. So this past summer, I did a field experiment where I went out and I collected pollen from all 3 sites, which we can see in this graph here. So Marin County is shown to produce higher amounts of viable pollen, which are the shown on the left in the green and yellow, or green and blue circle and triangle, and then Santa Clara on the right is the orange circle, which is shown to produce less viable pollen, and we can see that in this table right here as well. So pollen was collected in the field, inserted in centrifuge vials, and brought back to the lab where I stained it and then observed it with a microscope, so the color denoted if it was viable or aborted, being red versus blue-green. So as you can see in these maps, these are the actual GPS points of the specimen samples. So this is one of the Marin County sites, the other Marin County site, and then finally our Santa Clara County site. So the goal of this project is to see the environmental factors such as precipitation, temperature, wind speed, land coverage, and soil type, and create a site suitability analysis.
So we chose these factors after diligently searching multiple data websites or public websites, and we chose and went with the PRISM, Oregon State University's research, and we used their annual precipitation and temperature raster data sets. We also used ArcGIS Living Portal for national land cover and wind speeds, and then for USGS is geologic type layers. So here you can see this is the 2021 annual precipitation layer that we got from PRISM. You can see the raster is slightly coarser from the public data set. And once again, Marin has that wetter climate ranging here from 330 to 1200 between both sites millimeters per year for 2021. And then for wind speed, which is important for speed dispersal, we see here Marin has a slightly higher. The sites were at 3 meters per second versus Santa Clara counties were at 2 meters per second more inland. This was also from ArcGIS Living Portal. And then on the National Land Cover database from ArcGIS Living Portal, you can see here the raster is very much higher resolution, and both sites happen to be in deciduous forest and mixed forest land covers. Then looking at the temperature, we use 30-year average because if we're considering climate change, we're looking at the past, and these samples were currently taken in the summer. So here you can see Santa Clara County has that inland higher temperature, and we converted from Celsius to Fahrenheit with the expression. And then moving on to the geologic layers, you can see here Marin County has the serpentine soil. That's in the bright green to the left. And then in Santa Clara County, Serpentine here, and this one is in bright blue, and that's right underneath these sites. Then going into our model for our site suitability analysis, we use all of these raster layers and mask them using mask to clip them to the county boundaries. And then we use Euclidean distance from only serpentine soil, and then reclassified that to raster. And we reclassified all by using ranking by threes. And then we reclassified through a weighted sum analysis with serpentine soil most important at 40%, land cover 20%. They had the same temperature and wind, both at 10% respectively, and precipitation at 20%.
Moving forward, we can see our site suitability analysis. So here in Marin County, they range from 1 to 3, like Jeanette said. So 1 is highly suitable, 2 is moderately, and 3 is least suitable. So in Marin County, we can see that both sites actually fall in highly suitable areas, which is very exciting. In Santa Clara, we can see that they actually fall on a moderately suitable site, which is interesting to see from this table here. Santa Clara produced the lowest amounts of viable pollen, while Marin County produced the highest, so it actually does line up, which shows that our site suitability analysis proves that environmental factors do actually have an impact on the amount of viable pollen produced. And this is really helpful for my project because I'm doing 2 experiments in a controlled greenhouse and then 1 in the field experiment, so I'll be able to compare those results together. And then moving forward, we would like to do a statistical analysis to fully validate this hypothesis, because while we have good results so far, they're not statistically accurate or proven. So if we could purchase more intricate and detailed raster layers, that would be very helpful. That would allow us to zoom in on those exact GPS points to get a little closer. So we would like to thank both our professors, David and Fernanda, for leading this class and helping us with our project, as well as my professor, Dr. Sivan Suni, for overseeing my thesis preparations and fieldwork. Thank you.
Thank you.